Hello everyone, welcome back. So this week we're going to be discussing bone histology and bone just general terminology in lab. So this is kind of a precursor to the skeletal system. Next week we're going to be learning all of the bones and the features of the bones. But the way the features are named is based on some just general terminology. So this week we're going to focus on that general terminology. So then hopefully next week, when we get to learn the names of the different features, everything makes more sense. Okay, so we're going to start by talking about just bone basics and the histology, so different types of bones and what they look like. Okay, so there's four main types of bones. We have long bones, which we're going to see a lot of and are really important. Some examples are the femur and humerus. We have short bones, for example, our carpals, so the bones of our wrist are considered short bones. The bones of our phalanges are also long bones here. Okay, so long bones are going to be longer than they are wide. Short bones are a little nubbier. We have flat bones, for example, the bones of our cranium, the skull, and also our ribs are example of flat bones. And then we have bones that are irregular, such as the vertebrae, that don't really look like any of the other ones. Okay, so ones that are easy to confuse are long bones and flat bones because they are kind of similar. They're both, longer looking. But long bones are always going to be longer than they are wide. And a good way to differentiate between the two is long bones are always going to have a medullary cavity. So in the middle of the bone, you're going to have this hollow cavity. It's going to be filled with bone marrow, but flat bones don't have that, right? So flat bones on the inside have a layer of spongy bone, but they don't have that cavity, that opening in the middle. Okay, so obviously it's not super easy to tell what the inside of the bone looks like, but it's going to be pretty straightforward when we get to lab. You'll be able to see some long bones that are cut open so you can see that cavity. Okay, so inside the bone, all bones are going to have compact bone on the outside. So most superficially along the outside of the bone is going to be all compact bone. And then on the inside, you're going to have spongy bone. Okay, all bones do have bone marrow. So on long bones in that medullary cavity, that's going to be filled with bone marrow, like I said earlier. But bones that don't have that cavity do have bone marrow as well. And it's going to be housed in the compact bone. Okay. And there's two different types of bone marrow. We have yellow and red bone marrow. And the names kind of tell you what they're made of. So yellow is adipose tissue, and then red marrow is going to be forming the blood cells, and these is mostly going to be found in flat bones. Young children, as they're developing, are going to have more red marrow than fully grown adults as well. Okay, and then the spongy bone and flat bones, like I said, is kind of pancaked in the middle in all the flat bones. That's called diplo. I think that's how you say it. I've never said it out loud before right now. Oops. But that's the special name for the spongy bone in the flat bone. Kind of looks like an ice cream sandwich. So the spongy bone would be the ice cream or the diplo. Okay. So long bones have some more anatomy that you have to know. They all have a very similar structure, so there's fancy terms for everything. Epiphysis at either end, they're the ends of the bones, right? So here we have a femur, so there's epiphysis on the proximal end as well as the distal end. The diaphysis is the part in the middle, that's like the long shaft part of the bone. Okay, so that's going to be sandwiched in with two epiphyses. Again, we've already said this, but inside of that diaphysis is the medullary cavity that's going to hold the bone marrow. And the way we can differentiate between the epiphysis and diaphysis is that we have an epiphyseal line. So as we're growing, that's going to be the epiphyseal plate, which we'll talk more about in lecture. But once the bone's fully developed, that calcifies and becomes the epiphyseal line. Okay, so in all long bones, you'll see that line separating the two parts of the bone. Okay, and again, like all bones, you're going to have compact bone on the outside, and then in the middle, deep to the compact bone, is going to be spongy bone. Okay, and again, that spongy bone does have a bone marrow on the inside of it. Okay, so that's the basic anatomy of bones. We can also look at bones on a microscopic level. Just like all organs, all parts of the body, bones are made up of individual cells. So when we take them under a microscope, there's more stuff that you have to know. Okay, so the cells that make up bones, the majority of the cells in bones are called osteocytes. So they make up and maintain the matrix. So the matrix is all the stuff around the cells. So it includes a lot of different stuff, including collagen, which is just a protein, and calcium phosphate. And that adds some strength to the bones. Osteocytes, again, are the individual bone cells, and they reside in little cavities that are called lacunae. On the next slide, I'll show you what they look like. But I think of the lacunae as like little lakes, and the cells are just like floating in the lakes. Okay? And the lacunae connect to each other through little sprouts called caniculi. So I like to think of those as like rivers leaving the lake. So up here you can see the big area here is the lacunae. You have the cell or osteocyte in that lacunae, in that lake. And then leaving the lakes are the caniculi or the little rivers. Okay. We also have lamellae, which are layers of bone in both spongy and compact bone. And then osteons are your circular, typical bone structure. I'll show you in the next slide. Okay, and the lamellae, the osteons, are going to be around a central canal. Okay, so here is a model of an osteon that we have in class, and you're going to see this week. So again, the osteon is this whole round bullseye-looking thing, right? So it's made up of individual lamellae, which are individual rings of the bullseye, right? So you can see there's this outside ring, this is a lamellae, this next layer is lamellae, right? So each ring is an individual lamellae that makes up the osteon. In the middle of the osteon, you have the central canal, which holds blood vessels and nerves, things that are flowing through the bone itself. Okay, so this is on a microscopic level what bone looks like. Okay, and then if you go even further, you have the major structures. These little colorful things over here are the lacunae, osteocytes that we were talking about earlier. So between the lamellae, there's little gaps or openings, and those are called lacunae. So this lighter red area would be a lacunae. That's like the lake I talked about in the last slide. The lacunae holds the osteocyte, and an osteocyte, remember, is the bone cell itself. Okay, so we have osteocytes in a lacunae, and then the caniculi are the rivers that are going to be connecting the lacunae. Okay, hopefully that makes sense. But you're going to have these throughout the bone, throughout each osteon. There's going to be a number of different lacunae in different areas and caniculi that connect them all so that the cells can get nutrients from the blood and stuff in the central canals. Okay, and again in lecture you're going to talk way more about this. This is the only models we have to really show this, but you can see everything pretty well. Okay, so that's a osteon histology just on the model. You can also see this on an actual slide of a bone. Okay, so here you can see again the osteons, the big bullseye looking things. In the middle, we have our central canals running through, right? So over here, we'd have another osteon. We have the lamellae, which are the rings of the bullseye, if you will, right? Individual rings that make up the bullseye. The little dark circles are going to be the lacunae, right? And you can't see it, but each lacunae is going to have an osteocyte. Remember, the site is the cell. And then there's going to be little rivers running through the lamellae, and those are the caniculi, right? The rivers that are going to connect the lakes or the lacunae. In lab, you're going to look not only at the model, but actually at a slide of tissue of actual bone. So it's pretty cool, you can see that. All right, besides that, there's some other layers of bones that kind of just hold everything together. So on the very outside of the bone, we're going to have a periosteum, which is an outside like membrane. I can see here it's like peeled back. That's just connected by these perforating fibers. So that surrounds the bone. And then in long bone, or I guess in all bones, we have the endosteum, which is on the inside of the compact bone, separating the compact bone from spongy bone. Okay, so we can see that there. All right, so that's just the basic histology and like makeup of bones. The next part of lab this week, we're going to be talking about skeletal terminology. Okay, so before we get into that, just general bone functions. Obviously, it's going to give structure to the body, right? So we can stand up and move around, and we know we're not just noodles, right? It gives us some structure. But bones are really important as well because they allow us to articulate, right? We're not made of one solid bone. We have individual bones that connect to each other that allow us to move. Obviously, we need muscles to do that as well. But articulations are made up of bones. Okay, so we have joints all over our body, different types of joints, which we're going to cover in lecture, not lab. But the skeletal system is really important because it allows us to articulate, so it allows us to move. Okay. So the bones that are articulating or moving against each other, those surfaces that are going to articulate have to be smooth. Okay, so we're going to see that different surfaces of the bone are going to have different textures. Anytime you see a very smooth texture, that's telling you that's going to be an articulation. And again, if you're, moving, you don't want a really rough surface because it's going to cause friction, it's going to damage tissue. You want something smooth that glides really easily. Okay, so there's two main types of articulations, a condyle, which is a rounded projection. So this is the distal end of a femur. So we can see the medial condyle, you have a lateral condyle, and the condyles are going to be smooth, right? Because this part of the bone is going to be articulating or moving. Okay, we also have what's called a head, which is going to be the rounded end of a bone. So here we have our humerus and the scapula. The head of the humerus, which is this white part, again is going to be really smooth. It articulates in the glenoid cavity of the scapula. So when we move our arm, we don't want any rough patches on the head because then it's going to scrape and be probably pretty painful. Okay, so those are our two main articulating surfaces. And again, those are always going to be nice, smooth surfaces. Okay, so those are condyle and head. Bones also have other projections, but these projections are important in muscle attachment, so they're not going to be smooth. These are going to be rough, going to be bumpy, kind of irregular shapes, so that muscles can attach to them, right? A muscle is going to have a difficult time attaching to a really nice, smooth surface, so the texture of these projections allows muscles to attach. Okay, and there's a lot of different ones. They all mean different things. So you're going to see multiple of these on different bones. I'd recommend like making flashcards or something to really remember what each of these are. But we have a tubercle, which is a small, rough projection, a tuberosity, which is a large, rough projection, trochanter, which is a very large, rough projection, and it's on the femur, a spine, which is a very slender ridge. And in lab, we're going to look at all of these on the bone, so I'll just go through it quickly. We have a line, which is a long, narrow ridge. Crest, which is a ridge that forms the border of a bone. Epicondyle, which is a rough projection superior to a condyle. And then a process, which can be literally any shape. So they're all kind of vague, but they're all going to be textured projections that muscles can attach to. So for example, like epicondyles or rough projection. So if we go back to this slide, we have the condyles, right, which are smooth on the femur. We have the epicondyles on the edges. So those are going to be rough and bumpy where muscles attach, right? And they're going to be superior. This one's kind of not super superior to it, but superior to the condyles. Okay. So those are projections for muscle attachment. Again, we'll show you examples of all of them in lab this week. There's also other features of bones that are just little depressions or holes in the bone. So they're not projections, but kind of the opposite. So these are examples of those. So we have a facet, which is a shallow, really smooth indent, and that's going to be for articulation. So it's going to be just super, super shallow as a facet. A fossa is going to be deeper. Sometimes the fossa is for articulation, sometimes not. It just depends on which fossa. A meatus is a tube-shaped cavity, so it's a hole that goes all the way through the bone. For example, on your temporal bone, you have your external acoustic meatus, which is where your ear canal goes through your skull, right? So it's a tube hole through the bone. A foramen is just a small, like, kind of pinhole that goes through the bone. Alveolus is a socket for a tooth in the mandible. We're not really going to be talking about teeth, so we're not going to see that. And then a sinus is a cavity within the bone. So we know we have our sinuses in our face, or I don't know, what do you call that? I guess it's your face, hey? So the sinuses are just cavities in your bones in your face. Okay, so those are all the holes or depressions you're going to see. And then the last type of structure we're going to see on bones are sutures. And sutures are fibrous bands of tissues that connect to the bones of the skull. So all the different bones of the skull are going to be connected by sutures. And we'll see this in lab. But all the sutures have different names. But as we're developing as a fetus, those sutures are soft and malleable and allows our bones of our skull to grow. Okay, so our bones, obviously, we're not born with a head as big as it's going to ever get. So those sutures are soft and movable when we're little. And then once we reach our maximum head size, those sutures calcify and become hard and immovable. Okay, so again, you'll see that in lab this week. You'll also see a fetal skeleton, which is really, really cool. You can tell that the sutures on that skeleton have not hardened yet, so they're still soft. All right, so that's it for this week. I would recommend, again, making flashcards or something for these, but we'll see everything in lab, so don't worry too much about it. Okay, bye.